right now.
Hello. All right. If you are done with your trivia, please bring them up front. To Meredith right now, open your hands up. Awesome. Bring them up front, and we will grade them and announce the winner after our first talk. Speaking of our first talk, our first speaker is a graduate student in the Astronomy Department at the University of Washington. He does all kinds of awesome work, all about exomoons, and I can't wait to hear him talk about it. Please join me in giving a very warm welcome to Tyler Gordon. All right. Hi there, everybody. So, as Trevor said, my name is Tyler Gordon. I'm here to tell you about searching for exomoons. Alright, so first to start off with a little bit of a self-explanatory definition here. An exomoon is just what we call the moon of an exoplanet. So anything orbiting a planet that's not in the solar system, that's an exomoon. Now, starting out in the solar system here, we have in our solar system 19 moons that are large enough to be rounded under their own gravity. And many, many more if you include things that aren't large enough to be rounded. But so just here in our solar system, we have more than twice as many moons as we have planets, big enough to have shaped themselves into spheres. So extrapolating outwards from there, we have every reason to expect that there are a lot of moons out there in the universe, probably many more than there are exoplanets. So why are we interested in finding these things? And a bit of a spoiler alert here, we haven't found any yet, but we're looking. So why are we interested in it? Why are we spending so much time on this? Well, first of all, the most interesting thing for me is just the question of habitability. So could exomoons themselves be habitable? We're really interested in finding places out there in the universe where life could be, and exomoons are potentially one of those places. The second reason, though, is that a lot of people are really interested in planetary formation, how planets come into being, and the presence or absence of a moon can tell you a lot about how that planet could have formed in the way that that whole process took place. So there's a lot of people that would be interested in exomoons for that reason as well. And then the final reason is the habitability of an exoplanet. Whether or not a planet has a moon can tell us a lot about the prospect for that planet being habitable. And there's a couple ways in which moons come into play there. One way is that moons raise tides on the surface of a planet. And there's a lot of people who think that that having tides battering a coastline might have been really important for delivering nutrients to the ocean and for providing places for life to come into being and then develop here on Earth. And then the second reason is that a moon can have a pretty significant effect on the orbital characteristics of a planet, specifically on the obliquity of a planet. And the obliquity of a planet is what we call the angle that the planet's axis makes with the plane of the solar system. So the Earth is tilted about 23 degrees away from straight up, and that little bit of tilt is what's largely responsible for seasons on Earth. Now, out there in the rest of the galaxy, you can imagine exoplanets that have all kinds of different tilts. Some that are tilted far more edge-on towards their suns, and those ones that are tilted towards their suns that way are going to have much more extreme seasons than we have here on Earth. And what an exomoon can do is help stabilize these planets to oscillations in that obliquity. So the presence of an exomoon can keep a planet from tumbling back and forth onto its side and can insulate it from having really extreme changes in season, which is something that we think could be very bad for life. But I want to go back and talk about habitability because, like I said, that's the most interesting thing for me. Um, we're used to talking about habitability for exoplanets kind of in terms of this concept of the habitable zone, where we have the situation that interior to the habitable zone, a planet is going to be likely too hot to support life because it's too hot to have water on its surface. So that's where we'd have a planet like Venus, for instance, which is interior to the sun's habitable zone and has a surface temperature of about 700 Kelvin and no water on the entire planet. It's all been driven off by the intense solar heating. And then exterior to the habitable zone, we have planets that are likely too cold to support any life. Any water that exists on the surface is frozen. And then there's this region that's just right in the habitable zone where planets like Earth can exist with water on their surface, which we think is a prerequisite for having life. So that's the picture that we have for exoplanets. But what about exomoons? We have a bit of a different situation for exomoons. So here I have a picture of, this is Io, the innermost of Jupiter's large moons. And right at the top there, you can see a little bit of a blue plume. And what that is that's the plume of a sulfur volcano on the surface of Io. And Io is covered in these things. And the reason that Io has all of these volcanoes is that it is strongly affected by tidal interactions that it has with Jupiter. So as Io orbits Jupiter, sometimes it's very close to the planet in its orbit, sometimes it's a little bit further away. When it's close, it gets elongated a little bit into what we call an ellipsoid, and when it's further away, it relaxes into a slightly more spherical shape. And so all of that tugging back and forth, that's stretching it out into an ellipsoid, and then back into more of a spherical shape, and back and forth, creates a lot of friction in the interior of the moon. And that friction leads to heating, and that heat has to make it out somehow. So it makes it out through the surface of the moon and causes all this volcanism. So that's another effect that's in play for an exomoon. And that tidal heating can either contribute to or detract from habitability. And I'll tell you why. So here we have kind of a schematic diagram showing regions around a planet. So here in the center is some kind of gas giant planet. In this red and orange region, you're too close to the planet to be habitable. You have Venus-like conditions just due to the tidal heating, due to interactions between the moon and the planet. And then exterior to that, in this blue and green area, you have far less tidal heating, and you might expect a habitable exomoon to exist in that region. And these different colors represent different things, but there's another level of detail there that I won't go into now. But if this gas giant that our exomoon is orbiting around is in the habitable zone of its star, then no matter how far away we move the exomoon, it'll remain habitable. Even if we move it all the way out and unbind the exomoon from the planet that it's orbiting, then it just becomes a habitable exoplanet because it's still orbiting in the habitable zone of the star. So we get this concept of the habitable edge, where rather than having a zone, a two-sided zone that's habitable, we have an edge within which it's not habitable and outside of which it is habitable. But now let's imagine that we move this planet out of the habitable zone into a region where a normal exoplanet would not be habitable because any liquid water on the surface would be ice. But for our hypothetical exomoon, you can imagine a situation in which it's just close enough to this planet that tidal heating provides the extra energy that that planet needs to be habitable, even where the sun isn't bright enough to provide that heating for it. And that's something that we see in our own solar system, and I think that our next speaker will probably mention that, where some of the moons of Jupiter are kept liquid in their interiors by these tidal interactions. They're able to maintain a liquid interior where they otherwise wouldn't be able to. So where are these things? Why haven't we found any yet? This is my mystery exomoon. Haven't found any yet. Where do we find them? And why are they so hard to find? So the main problem with finding exomoons is just the size of the things. We expect them to be really, really tiny. And the reason for that is that the mass of a satellite generally scales with the mass of the planet that it's orbiting. And that comes out of both observations of our own solar system. We can add up the mass of all the satellites of Jupiter, all of the satellites of Saturn, and compare those to the masses of their planet. And they tend to follow this rule. And it also comes out of planetary formation models. And it's the same rule. And the rule is that for a given planet, here I have a planet the size of Jupiter. We expect that the total mass of its satellites add up to between about 1/10,000th and 2/10,000ths of the mass of their host. And so here's Earth for scale, and here's about what that rule gives us for the mass of all of the moons of Jupiter altogether. So that's a bit bigger than our own moon, but not a lot bigger. It's a bit smaller than Mars. So if we move on and say we want to find an exomoon that's as big as the Earth. We found exoplanets the size of the Earth before, so maybe this is a reasonable thing that we can see one of these exomoons that's the size of the Earth. So if we have an Earth-sized exomoon, how big a planet do we expect that to be orbiting according to these formation models that we have? And the answer is that we expect it to be orbiting something that's about 30 times the mass of Jupiter. And at that point, we're not really even talking about planets at all. Something that's 30 times the mass of Jupiter would probably be a brown dwarf. So if you're orbiting a star, if your exomoon is orbiting the star, then maybe it's not properly called an exomoon at all. So the moral of the story here is that exomoons are probably really small, and small is a problem because small things are really hard to see. All right, now I have an animation I'm going to show you, and this is just a regular exoplanetary transit. There's no moon here, but this is showing along the bottom the flux of the star over time, and here's the planet. It's transiting in front of its star, and we get this dip when the planet is blocking out the light of the star, and then we go back to seeing the full flux of the star. Now here's what that picture looks like when we have an exomoon transiting along with its planet. We get kind of this double dip, where the exomoon transits and we see a small dip, both the planet and the moon, just the planet, and then we go back up to the full flux of the star. So we see kind of this overlapping transit effect. But what I should mention here is that I've greatly exaggerated the size of the transit due to this moon. In reality, we see something more like this, or we would expect to see something more like this. It would be great if we saw something like this. But, so this is with a Neptune-sized moon, which is a lot bigger than what we would expect for a Jupiter-sized planet. I showed you before that for a Jupiter-sized planet, we expect something just a bit bigger than Earth's moon. But what we have here is a Neptune-sized moon. So even this is greatly exaggerated compared to what we would expect to observe. And here you can barely see, right there is the start of the moon transit, and here's where the moon moves out of transit, and we're just seeing the planet. So this is that overlapping transit. And you can barely recognize it with your own eye, even at this point. Now I'm going to go back to the planet-only transit for a second and talk about what we can do to make this curve look a little bit nicer. Sometimes these transit curves don't look this nice. It's really hard to detect the transit by eye. So if we want to actually find out if there's a transit, one thing we can do is observe that star for a long time, observe multiple of these transiting events, and then stack them all up on each other. And then we can draw a line through all of those stacked transits, and we can get a much nicer, smoother transit curve. So that's something that helps us to detect these things in the first place, is to do what's called phase folding, which is where you cut up the light curve into these individual transits and then stack them on top of each other. Now, what happens if we do that for a moon? Well, the thing with a moon is that because that moon's also orbiting the planet, the moon and the planet aren't always in the same position when they transit in front of the star. So sometimes you get the moon's transit happening after the planetary transit, sometimes you get it before, sometimes it's overlapping to various degrees. You get the moon's transit in all sorts of different places at different times. So when we stack those, and we're going back here to the more realistic Jupiter-sized planet with a Neptune-sized moon, which is hard to see to begin with, and then we stack those, and we get kind of a coherent adding of the planet's transit. But notice the moon transits are obscured because they happen in different places, so we don't get that coherent adding of the exomoon's transit. But what we do get is a little bit of an increase in the scatter of these points just before and after the transit, and that's because of all of those little teeny moon transits overlapping. So we get this increase in scatter, and that's been referred to as the orbital sampling effect. And that's one thing that can be used to try and detect exomoons, just by looking for that signature. So that's exactly what a team of researchers did, led by Alex Tichy and David Kipping at Columbia University. They took a whole bunch of Kepler planets, hundreds of them, and they tried to find that orbital sampling effect for each of these, and they plotted something they called it the significance of the orbital sampling effect. And they plotted that. They made a histogram for all of these planets that they studied, and if there were no moons in that sample that they studied, you would expect to see this nice bell curve, like that, centered on 0, which is largely what they saw, except for this one outlier at a relatively high significance, and that's Kepler 1625b. So now we have an exomoon candidate that they're calling Kepler 1625b1, and we have several. Here are 3 individual transits. So these aren't stacked transits; these are individual transits of Kepler 1625b. And they plotted those with a model of both the planetary transit and the exomoon transit. And I don't know about you, but if I look at this by eye, it's not super easy to see that there's a moon here. I can kind of see, in this one a bit of a dip there, and in this one it does look like there's a dip on either side of the transit. Not totally clear all the same though. But they are following that up. In October of last year, they observed with Hubble. So they're going to take a closer look with a bigger telescope and see if they can confirm that there's an exomoon there. Another thing to notice, though, is that Kepler 1625b is thought to be, they say, about 10 Jupiter masses, which is really, really big for a planet. That's about on the edge of what we would start to consider a brown dwarf. So there's a couple questions still here that need to be answered, and hopefully will be answered. Maybe have been answered already since they observed this thing, but that data isn't out yet. So I don't think anybody besides them really knows what they might have found there. So the questions that remain are, is Kepler 1625b even a planet if it's that big? Is the moon actually even there at all? That's what Hubble will hopefully answer. And then, if it is there, how can such a large moon form? This moon, from those transits, they were able to derive a radius of the moon that is about the size of the planet Neptune. So that transit that I showed you earlier, a Neptune transiting a Jupiter-sized object, that's kind of what they've observed here. And that moon is a lot bigger than what we would expect according to that scaling law, where a moon is about 1/10,000th the mass of its planet. This would far exceed that. So there's kind of a question there about how such an object would have even ended up in orbit around this planet. So we will hopefully sometime in the coming months find out what they saw with Hubble and find out whether there actually is a confirmed exomoon or not. Okay, and just briefly to wrap up here, I want to talk about the future of searching for exomoons. And I put up here a picture of JWST, which we often do when we're talking about the future as astronomers. JWST is going to be helpful in finding exomoons, not because it has like a greatly better precision than Kepler. Kepler was optimized for looking for those transits. JWST isn't going to do necessarily a lot better than that, but what it does do is have a lot better wavelength coverage. Kepler only looks in one wavelength band in the optical, so it doesn't see that dependence on wavelength. And what we expect for exomoons is that these things are small enough that their transits are going to be largely hidden by the variability of the stars. And we think that we can use the wavelength dependence of stellar variability. So stellar variability depends on wavelength, whereas the transit of a moon in front of a star does not depend on wavelength. So by using JWST's ability to see in different parts of the electromagnetic spectrum, we may be able to disentangle the transit of an exomoon from stellar variability that could obscure that transit. So that's what we're looking forward to. Are there any questions? Yeah, right here. Let's start there. Yeah. So the question was, does I showed those pictures of the habitable edge for an exomoon. If I can get back to that, that is this picture, right? Uh, so the question was, does this picture depend on how the exomoon rotates? Does it matter if the moon is tidally locked to the planet or not? Um, I would say mostly not. In the case of an exoplanet, it matters whether it's tidally locked or not, because if it's tidally locked, it has one face on to the star the whole time, which means that it's only getting heated on one side. But this planet isn't providing hardly any heat to the moon itself. The heat that is due to the tidal interaction between the planet and the moon is coming from the interior of the planet. So it won't depend very much on how the moon is rotating. What it does depend on are things like the eccentricity of the exomoon in its orbit, how circular that orbit is. An orbit that is less circular will experience tidal heating, and the zones will move outwards in that case. Next question? Yeah.
So, how do you know the relative mass? You only know the relative size, so it depends.
Yeah, the question was, how do we know the relative masses of the planet and the moon? And the answer is that we can't get that from observing a transit. There's no way to know. All that gives us is the relative radii of the planet and the moon. What we can do to get the masses is we can look at, if we do ever manage to observe one of these things, we can look at their gravitational interaction through the way that the gravitational tug between the planet and moon changes the exact timing of the transit. And that's called transit timing variations, and that can give us some insight into their masses. Okay, the question was, what is stellar variability? So yeah, I didn't define that very well when I was talking through my last slide, but stellar variability is how much the star changes in brightness by. So all stars, even stars that we consider to be not very variable, change slightly in brightness. And one of the things that can cause that is stars tend to have spots on them. We see sunspots on our own sun, and those spots move across the disk of the sun, and when they do that, they cause a slight dip in the brightness of the star. Stars also flare. The surface of the stars are covered with convection cells, little what look like granules, and the changing pattern of those granules causes variability in the brightness of the star. So all of those things can happen. Yeah.
If it turns out that the real example that you gave that was observed happens to be an exomoon, at what point do you have to kind of redefine what the mass difference between like a moon and its thing it's orbiting, what make it qualify as a moon versus like a binary planet?
Yeah, good. The question there was, if it turns out that we start to see like really, really large moons orbiting their planets, do we have to at some point consider like our definition of a moon? What's the difference between a moon and potentially like a binary planet? And I don't totally know the answer to that question. Some people will probably come up with definitions. I think one kind of easy definition. I think this has been applied to Pluto. For instance, Pluto has a moon, Charon, that is really, really large compared to Pluto. It's not a lot smaller than Pluto is, and they actually orbit a mutual center of mass that is outside of either body. So I think that's one easy definition, is to say if the center of mass of this system is outside of either body so that they're orbiting each other rather than one orbiting the other, then you might tend to call that a binary planet rather than a moon. One more question. Yeah.
What about the edge cases like binaries or you have to look at it.
Sorry, can you say that again?
Edge cases like binaries we just talked about, or antimatter planets, or stuff like that.
Yeah. So the question there is, what are the edge cases, like in the case of a binary planet, or you said antimatter planets. I don't expect that we will find any antimatter planets. I think that they would annihilate with regular matter planets. They probably wouldn't have been formed in the first place, because we expect that there's a lot of regular matter around, and all of the antimatter has at this point been annihilated. But yeah, the edge cases are in my mind, a moon that is almost as big as its planet, such that they're orbiting each other. That would probably be a binary planet. Another edge case that kind of came up was what about when the planet gets to be too massive to be a planet when it's a brown dwarf? And that's kind of an interesting case because a brown dwarf is a bit different than a regular star, right? So finding something orbiting a brown dwarf is still pretty exciting. And I think at that point, the question of whether we call that a moon or a planet in a binary system is a bit more interesting. I don't know the answer to that.
Thank you so much, Awesome. Wow, awesome stuff. All right, so before we get to our next speaker, I have to announce who won trivia as well as give you guys the answers were. So. All right, I hope everyone's had a chance to get themselves a new beer, get themselves a little cycle dogs. Everyone's doing great. Our next speaker is also a University of Washington graduate student from the Department of Oceanography. He's going to be talking about some awesome things, so please join me in giving a very warm welcome to Max Showalter.
I've never given a talk before after a few beers, but it's kind of nice. So, like you said, my name is Max Showalter, and I'm an oceanographer, not an astronomer, so first disappointment of the night. So for the next couple hours, it's going to be oceanography on tap, not astronomy on tap. Got a strong oceanography contingent in the front. I work in a lab that studies bacteria and viruses in extreme environments, in polar environments and sea ice, and how they adapt to those environments. And then from there, what can we learn about those organisms and how we might find life in outer space in similar environments? I really like this picture because I think it looks like one of those wish you were here postcards, except I don't think anybody would wish they were here. But here we are, and it's a beautiful day. It looks sort of like a scene from Fargo, even though it's up in northern Alaska, but you might expect maybe there's a wood chipper off to the side over there or something. But in this picture, we're about to go into this box here, which is actually a tunnel into the ground, into the permafrost, to look for some microbes in frozen soils from 10,000 years ago. But I'm not going to talk about that today. I just wanted to reference Fargo. So today I'm going to talk to you a little bit about a project that I did with some folks from Caltech, collaboration with people at Caltech and NASA JPL, to look for a new way to look for life in outer space. But I'm going to start with a story that some of you guys might be familiar with. I read this book a lot when I was a little kid, and some of you guys might have too. It's called 7 Blind Mice. So it's about 7 blind mice, as you might expect, and they live next to a pond. And one day they realize that there's something there that they've never experienced before, and they're trying to figure out what it is. So they decide every mouse will go out on a different day and try and figure out what they can learn about this thing and figure out what it is. So on the first day, the red mouse goes out, and he goes out there and he's walking around and he feels something that's like really strong and really tall, and he decides that it has to be a pillar. And he walks back and he tells the other mice, and the mice say, no, it's not a pillar. That's stupid. It's not a pillar. So they send out another mouse. And on the next day, the green mouse goes out, and he feels something that's really long and wriggly, and he decides that it's a snake. And the other mice say, obviously it can't be a snake, because the first mouse said it was a pillar, and also you probably would have been eaten if it had been a snake. So then they send out the third mouse, and the third mouse says, I felt it, it's really sharp, it's a spear. Comes back, and none of the mice agree with him. And so this goes on, and the mice say, it's really steep, it's a cliff. Or I felt the wind, it's a fan. Or I pulled it, and it's a rope. And all the mice are arguing and arguing about what it could be. It's a fan, it's a snake, it's a cliff, and none of them can agree. So that's when the seventh mouse goes out, and he walks all up and down this structure, and he feels everything with his little mouse whiskers, and tries to figure out exactly what it is. And he gathers all this data, and he comes back, and he says, all right, guys, I felt it, and it is indeed as tall and strong as a pillar. It's long and wriggly like a snake. It's sharp as a spear. It's got steepness like a cliff. It moves like a fan, and I can pull it like a rope. It's an elephant. And then all the mice agree, and they all live happily ever after, I guess. I guess this isn't like Dumbo, where elephants are afraid of mice. But, okay, so why am I telling this story? I think there's a lot of things that we can learn from this story as astrobiologists when we're looking for life in outer space. And the first thing is that it's really easy for us to see exactly what we want to see, even if that's not what's there. Just like the mice are seeing the pillar or the snake, that wasn't what's there, but that's what they got based on the information. And the second part is that we need multiple tests in order to identify what we can't see. So it wasn't until one mouse had collated all that information, different ideas, that they finally figured out what it really was. So I'm going to call this the 7 blind mice principle. I think it's really useful for astrobiology, and to my knowledge, no one else has used this story in the context of astrobiology before. So I'm going to go ahead and put that up there. Maybe I'll say that for me. So you heard it here first. So as astrobiologists, we're looking for biosignatures, which are just hints that there might be something alive or something that has been alive in the past. And there's a lot of examples of this. So one example are chemical signatures. So you might think of plants that photosynthesize, they produce oxygen. You know, if you see oxygen in a place where you wouldn't otherwise expect it, that might be a really good indication that you might have found life. Obviously, if we see a tree, we can all go home. Another example are structures, like fossils or something. So here's a picture of the Stromatolite, which is sort of like a fossilized bed of bacteria. So these bacteria grow on top of each other in mats. And over time, like sediment grains and other stuff gets entrained inside of these mats, and then it all gets pressurized and fossilized. And you get these really unique structures that, to people who study rocks, this is really obviously life. I wouldn't be able to say that, but to people who study rocks, this is really obviously life, or it had been life in the past. Another example is biological molecules. So things like amino acids, which are the building blocks of proteins, or nucleotides, which are the building blocks of DNA. They tell us that either life has been there in the past, or life is there now, or life could be there in the future. And just like with the elephant, we want lots of biosignatures all telling us the same thing in order for us to decide that there's life. But sometimes as human beings, there are biosignatures that are so obvious to us that even when life is right under our nose, we don't see it until this happens. And so an example of that is movement. And so the project that I worked on with a group from Caltech is taking this to a more micro scale and proposing movement as a new biosignature. So bacteria use movement. When it's bacteria, we call it motility, which is basically just bacterial swimming. And so the idea here is that we can go out into space and look for bacteria swimming, which is sort of such an obvious sign that something's alive that we could consider it like a waving person in the crowd. Like we're looking down at the sample and someone's waving back at us, like, hey, we're here, you found life. So here's an example of some bacterial motility. This is from some Antarctic bacteria. And you can see how vigorously they're swimming. It's kind of cool how fast they are. This is real time. But when we're thinking about motility as a biosignature, we have to ask ourselves two questions. First of all, will things actually swim in space? It's a valid question. If they're not swimming, we can't use this as a biosignature. And if they do swim, how are we going to see that? It's hard enough to see microbes on Earth, let alone millions of miles away. I think it's easiest to answer these questions in the context of where we're thinking we might find life. So as people who want trivia might know, Europa is one of the most obvious places where we might be looking for life in outer space. So here's a picture of Europa. I think it's really beautiful to look at. Europa is one of these places where we're most likely to find life because we know that it has this liquid ocean inside on the moon, covering either most, if not all, of the moon. And furthermore, we know that it's salty. And so I kind of expected Tyler to talk about why we know that. So you're just going to have to leave in mystery because I didn't prepare that part. I don't know why. It has to do with conductivity, but you should go Google that. It's your homework. We also expect that there's lots of hydrothermal activity happening on this moon, which gives a lot of energy and maybe also redox reactions to the microorganisms that might be there, providing a source of sustenance for these microorganisms. But one of the challenges is that there's this huge icy crust on top. So obviously it's cold in a moon that has no atmosphere. So we have this 4 to 8 kilometer thick crust of ice on the ocean of Europa. And so for comparison, in the Antarctic, the maximum thickness is about 4 kilometers in the ice sheet. So it could be up to double that. So we might ask ourselves, so could something live here in this salty ocean with an icy crust on top? So to answer that question, we can think, well, we have one of those salty, crusty oceans too. An example of this is in the Arctic. So obviously we know that our ocean is salty, and in the Arctic and the Antarctic, it freezes every year. Maybe not as much as it used to, but there's still some ice up there. So here's a picture just to orient you. Here's Alaska, and here's Russia. And so it's a really good analog for places like Europa. We can go there and learn more about what it might be like on Europa. But you might ask yourself, do things live here? So to answer that question, we're going to zoom in on this spot right here. So this is Utqiagvik, Alaska, formerly known as Barrow. So it's the northernmost point in the northern hemisphere, or in the United States, not in the northern hemisphere. And it's where a lot of field work is done for American polar scientists. So the lab that I work in goes there fairly commonly. And so here's a video from underneath the sea ice near Barrow, Alaska. And so you're looking around. One thing you'll notice is that the bottom of the ice looks really brown. And you might think, well, there's a lot of dirt in that ice because it's close to shore. But actually what you're looking at is algae that are living on the bottom of the ice, and they're growing to such intense biomass that you can actually see them with your naked eye, even though they're microorganisms. In addition to being extraordinarily beautiful, sea ice is really unique because it's made from salt water. So when salt water freezes, there's two things that can happen to the salt. Either the salt can fall out of the bottom of the ice in a process of what's known as brine rejection, and that happens because the salt doesn't get crystallized into the ice itself. So it gets pushed out of the solid ice and falls because it's more dense. Or it can get stuck inside the ice if it doesn't have a chance to fall out, and that forms what are called brine ports. So when the salt gets stuck inside of the ice, the salts get concentrated by all this ice growing around it. And just like when you throw salt on an icy driveway and that melts the driveway, the salt inside these brine pores lowers the freezing point of the liquid so that you get these liquid pockets within the ice, even though the ice around it is frozen solid. And you can visualize this pretty easily. So here's an example of a freshwater ice block and a sea ice block. And the first thing you might notice is that the freshwater ice is really clear, and the sea ice is really opaque. It's really bubbly looking, right? And that's because of the brine pores. So the brine pores inside of it are obscuring the light getting through. And sort of a fun experiment that you can do at home if you have kids, or I guess if you don't have kids, you can also do it, is if you drop some food coloring on top of these ice blocks, food dye, on top of the ice blocks. On the freshwater ice, it just runs off the side. But on the sea ice, because there's these brine pores, it'll actually soak in and dye those brine pores themselves, so you can see the difference. So these brine pores are great because they make a really good habitat for a lot of things to live inside the ice. So here's an example of that. What you're looking at here, it's kind of hard to see maybe in the back. Here's ice, and then up here is the brine, and these are actually algae inside the ice, diatoms on the micrometer scale. There's also bacteria in there. You can't see them, but you'll have to take my word for it. So we can ask ourselves, okay, we know things live in ice. They can grow, they can metabolize inside of ice, but are they actually swimming in ice? Well, before we answer that question, we'll say that we know things can swim in the lab in conditions similar to that of ice. So what you're looking at over here is a video in real time of a bacterium that was taken out of sea ice and then subjected to conditions similar to that. So this is negative 10 degrees Celsius, which is about 15 degrees Fahrenheit, and that's the world record low temperature for bacterial motility. So get excited. And this was taken by a student that worked in our lab in 2002, which you don't think about it, but that was an incredibly long time. So this poor student had to sit inside of a freezer room up in Fairbanks, Alaska for hours at 15 degrees Fahrenheit, working on this special factory-modified microscope that worked at sub-freezing temperatures to get this data. And over here, I included some still shots because I wanted to highlight how fast this bacterium is moving. So these are speeds at -5 and -10 degrees Celsius. And at -10, the bacterium is moving at 28 microns per second. So a bacterium is about 1 micron in length. So this is 28 body lengths per second, which is about the equivalent of an adult man running half a football field in 1 second. So these guys are moving really fast at low temperatures. They're really good at swimming at these temperatures. Okay, so this is what happens in the lab, but do we see motility within sea ice itself? So let's start by putting up a little diagram. I'll do this myself. Here's ice and seawater underneath. One way that we can get to the ice itself is by taking a core. So we take an auger and we drill into that ice and we take out a chunk, take out a core. And this is a very intense process that requires exactly 5 people. And then you pull it out of the ice and it requires exactly 6 people. And I just want to highlight, down here you can see this algal band, maybe not the people in the back, but you can see the algal band of algae living inside the bottom of the ice and growing to really high biomass, but that's an aside. Okay, so you can take out this ice core, and then you can either cut it up or do whatever you want to it and take it back to the lab on your snowmobile, yada, yada, yada. Another thing that you can do, though, is drill an ice core that doesn't go all the way down, so it's not going into the ocean yet. There's still some ice in the bottom. And what will happen is that brine from the ice, from those brine pores, will drain into it because of the hydrostatic pressure. And this is called a sack hole, and I don't like that word any more than you do. But it's really useful because it allows us to get brine at near in situ temperature when we need it. You have to wait maybe an hour or two depending on the temperature. But the problem with this is that you get this sack hole brine, and then you take it back to the lab, and then you put it under a microscope, and this could take hours to days depending on where you're getting this ice. And so every minute that you, or every second that you have the bacteria out of this ice, out of the brine pores, or in different conditions, it changes what they're experiencing. And so you're not really sure if what you're seeing in the lab anymore is what's happening in the ice. So we can't use a microscope out on the ice. Or can we? So the project from Caltech is that they've built this microscope called a submersible holographic astrobiology microscope with ultra resolution, which is a really long name, but anybody that's read anything about NASA knows that they have the best acronyms. So this is called SHAMU. Here's a picture of SHAMU. Sticker included. So this orange box here is the microscope sitting inside of sea ice in Greenland. And these two orange balls in the back are just so it doesn't fall through and sink and ruin millions of dollars of grant money at the bottom of the ocean. But this microscope is about that big, maybe a foot and a half. And it works on principles of holography. And like I said, I'm an oceanographer, not an astronomer, so holography is not my strong suit. This is for the engineers. But I'll try to explain it briefly. So what happens is, inside of that orange box, there's a laser source. And that laser source splits into two beams. And one beam goes through your sample. So in this case, our sample is brine that may have swimming bacteria in it. And another laser beam just goes straight to the camera. And what you get are two waves of light that interfere with each other. And you can put that into a computer or whatever astronomers do and turn it into holograms. So 3D images of a cube of liquid that you can see bacteria swimming in. And so one of the most impressive things about this microscope is that it has resolution down to 0.8 microns. So remember, a bacterium is about 1 micron. So you can see bacteria swimming inside this fluid. Some caveats. It's still in development, so far, you actually still need to hand load the sample in. So you get near in situ temperatures, or near in situ conditions, so we can take the microscope out to the ice, take some brine out, and put it right into the microscope, which is still better than bringing it all the way back to the lab. But it's not quite to the state where you can just sample something and have it all automated. But it's better than that. So here's some we took it up to Greenland in 2015. So we took it down to Nuuk, which is in the southwest corner of Greenland, and here's an example of what it looked like. Pretty sure that's me. And so we're coring the ice like I was telling you about. Here's a picture of me meat mugging with the microscope out on the ice. And this is sort of the data that we get. So you can see this organism swimming in the brine that was pulled directly out of the sea ice at -4 degrees Celsius. So you might say, okay, so we can see things swimming in brines on Earth, but what if they don't swim? What if we get all the way there and they don't swim? Well, we have ways of making things swim. But nice ones. So we give them something that they want. So bacteria like amino acids, so the building blocks of proteins. So here in this example, we're giving these bacteria serine, which is an amino acid, which is basically really good food to them. So they eat this food, and you can see in this video they're like swimming this direction, and they sense this gradient towards serine, and they just change direction and start swimming up towards it. So organisms that might not have otherwise been swimming will swim by this process known as chemotaxis, which is just swimming in response to a chemical gradient. And sort of as an aside of this project, we actually set the world record low temperature for observed chemotaxis in a bacterium at -8 degrees Celsius. Makes me feel a lot better about all that work. Okay, so let's put this back into context. So we're talking about looking for life in Europa, right? So what does all of this mean? We can see things and sea ice on Earth, but what does that tell us about life in outer space? So one thing that I didn't show you or talk about much on that last figure of Europa was that we have these plumes coming out of the moon. So it's like a geyser of water shooting out off the surface of the moon, or off the surface of the moon. And we're pretty sure that this water is sourced from the ocean underneath, which presents a really incredible opportunity for us as astrobiologists or astronomers, if you're one of those people, to be able to sample that ocean without having to drill through 8 kilometers of ice. So you can imagine now we might be able to fly through those plumes, collect some of the material, and then use multiple lines of evidence, just like the mice, to look for life. And this is sort of similar to what's planned on the Europa Clipper mission in 2022 to 2025. They've already chosen all the instruments, I think, for the Europa Clipper mission, and there's none that are looking for motility, but we have hope for the future that motility might be a useful biosignature in future missions towards icy moons. So to close out, I just want to remind you that even as blind astronomers or astrobiologists or mice, we can identify an elephant sometimes if we have all the right information and we're using all the right tools. Thank you. Any questions? So the question was, what is the metric that we use for motility? So once the samples are reconstructed, we can use a computer algorithm to track the bacteria, and from that we can get velocities, changes in direction, especially in the background. So for example, if we have a sample that we spike with some sort of like small bead that's the same size of the bacterium, we can see if there's any flow that's happening and then compare the bacterial movement and
Is there any consensus as to whether life would be more likely to develop on the surface or an environment without light, like the bottom of the oceans?
Okay, that's a good question. So the question was whether instances where life might develop on the surface of the ocean or like the bottom of the ocean. What's more likely? So that, I think, sort of depends on the environment. Obviously, we don't know how life originated up there yet. I have a lot of ideas, and one of the ideas is that it might have originated at hydrogen slopes, like the molecules. And the reason for this is there's lots of redox gradients down there, so lots of different metals, different gases that they can use to stabilize. Also, it's going to form the hydrogen events. So if you think about a place like Europa, it might have these hydrogen events. It's also a good idea that there might be a lot of life there. So the question was if I had to guess with the Europa Clipper find life. My guess is no. But I don't know if that means that there won't be life there. I think that what I think would happen, the most likely scenario is that we'll find a lot of clues that might indicate there is life, but nobody will be able to rule out if there is life or not. So it'll sort of be ambiguous there. I'd like to think that there's life there. Any questions? Good, thank you.
Thank you so much, Max. That was awesome. Thank you all for coming out to another Astronomy on Tap. We will be here next month, February 16th. That is a Friday at 7 p.m. Please join us then. We'll see you then.